If your blood type ends in a minus sign, most people assume you have something unusual in your blood, something added, something extra. You don't. You're missing something. As though something reached into the human blueprint and took a piece of it away, and then handed the rest of the book to the next generation, as if nothing had happened. And here is what stopped researchers cold when they finally mapped it. The same missing result shows up in Africa. It shows up in Asia. It shows up in populations that have been separated from one another for tens of thousands of years. But it arrives there by three completely different mechanisms, three different genetic accidents, three different roads to the same destination. Whatever happened here, it did not happen once. It's separately, on separate continents, to separate people, at separate moments in the deep past. And the reason it survived at all, the reason it wasn't erased from our species long before recorded history began, is a question that took science more than 80 years to even begin to answer. Some of it, they still can't answer. Most of us barely think about it. It's a letter and a symbol on a card in a wallet, a line on a hospital form, something a nurse writes down before a surgery, or something you learn once when you donate blood and then forget for the next 30 years. A plus or a minus, positive or negative. Two words that sound like a grade on a test, or a mood, or battery terminal. Nobody explains what they mean. Nobody tells you that the minus sign is not a measurement of quality. It isn't a warning label. It's a record of an absence. And that absence, it turns out, is one of the strangest things written into the human body. A piece of missing text that carries information about who your ancestors were, where they survived, and what they lived through, long before anyone was writing anything down at all. So let's start with what it actually means. Every red blood cell in your body is covered in proteins, thousands of them. They sit on the outer membrane like markers on a surface, and your immune system reads them constantly, checking each cell, asking a very simple question over and over again. Are you mine, or are you a stranger? The A and B system that gives us the familiar blood types is one set of these markers. The rhesus system is another, and it is much larger and much more complicated than most people realize. There are dozens of separate antigens inside it, more than 40 additional markers beyond the famous one. But one of them dominates everything, because one of them is aggressive enough to matter. That's the D antigen. If your cells display it, you're positive. If they don't, you're negative. That's the whole difference. One protein, present or absent. The story of how we found it starts in the late 1930s in a hospital in New York, with a woman who had just lost a baby. She hemorrhaged during the delivery and needed blood urgently. Her husband was the same familiar type she was, so his blood was used. And she reacted violently to it. That should not have happened. By everything medicine understood at the time, that transfusion should have been perfectly safe. Two physicians, Philip Levine and Rufus Stetson, studied her case and realized they were looking at something that wasn't in any textbook. Her body had learned to attack a marker that her husband carried, and that her lost child had carried, and that she did not. Around the same time, Karl Landsteiner, the man who had discovered blood types in the first place and won a Nobel Prize for it, was running experiments with Alexander Wiener using blood from rhesus monkeys. They found an antigen there that also appeared in most human beings, but not all of them. The two lines of work came together, and the marker got its name from the monkeys used in the laboratory. Rhesus, later shortened to RAH. It's worth saying plainly, because a lot of confused ideas have grown out of this one accident of naming. The Rh factor is not called that, because Rh-negative people are less related to primates, or more related to them, or anything of the kind. It's named after the animals in a laboratory in the 1930s, that's all. A historical footnote that became a permanent label. But the naming was the easy part. The hard part came decades later, when molecular biology got good enough to actually go and look at the gene. And when researchers went looking for the RhD gene in Rh-negative Europeans, they found nothing. Not a broken version, not a fragment, nothing at all. The gene sits on the short arm of chromosome 1 in a crowded neighborhood of DNA, and in Rh-negative individuals of European ancestry, that stretch of the sequence is simply empty, a gap where the instructions should be. Both copies, inherited from both parents, missing the same piece. That raised an obvious question. How does an entire functioning gene disappear from a chromosome? DNA isn't supposed to lose whole paragraphs, and the answer, when it came, was elegant and slightly unnerving. The RhD gene is flanked on both sides by two long stretches of DNA that are almost identical to each other. Each one runs about 9,000 base pairs. They match each other by roughly 98.6%, and they run in the same direction. Researchers name them the rhesus boxes. Think of them as two nearly identical bookmarks placed on either side of a chapter. Now here's the problem with nearly identical sequences sitting close together on a chromosome. When cells copy and recombine their DNA, they line up matching sequences before making the exchange. It's a matching process, and it's usually reliable, but when two long stretches look almost exactly the same, the machinery can line up the wrong pair. It can match the bookmark on the left to the bookmark on the right. And when it does, and the exchange goes through, everything sitting between those two bookmarks gets snipped out and lost. The chapter disappears, the book closes again, and the two bookmarks fuse into one, and the chromosome looks perfectly intact. Except an entire gene is gone. That's it. That's the origin of Rh negative blood in Europe. Not a mutation that added something, not an alteration that changed a protein's shape. A clean surgical deletion caused by the DNA's own repetitive architecture folding on itself in the wrong place in one person at one moment, probably tens of thousands of years ago. And because that deletion is permanent, because you cannot recover a gene that has been removed, every single one of that person's descendants who inherited the deleted version carries the same silence in the same place. It is one of the tidiest examples in all of human genetics of how a single ancient accident echoes forward through thousands of generations. If the story ended there, it would be interesting but simple. One event, one lineage, one clean explanation. But then researchers started testing populations outside Europe, and the neat picture fell apart almost immediately. In African populations, Rh negative blood exists too. Not at the European frequency, but at around 8%, which is far too common to be an accident of migration. So the obvious assumption was that the same deletion had traveled there. It hadn't. When geneticists tested Rh negative donors of African descent, they found that in about 2/3 of them, the Rhd gene was still present, still sitting exactly where it belongs on chromosome 1, fully in place, but it doesn't work. Inside the gene, in the 4th exon, there's an insert of 37 extra base pairs. That insertion throws off the reading frame, the way the cell reads the sequence in groups of 3, so everything downstream comes out as nonsense. And to make it final, there's a premature stop codon sitting in exon 6, telling the cell to quit reading long before the protein is finished. The instructions are there. They're just unreadable. Geneticists call this kind of thing a pseudogene, a ghost of a gene. It looks alive from a distance and produces nothing at all. Of the remaining African Rh negative individuals, about half carry the same deletion Europeans have, and about half carry something different again, a hybrid, where part of the Rhd sequence has been swapped with sequence from its neighboring gene, producing a scrambled version that makes no D antigen at all. Then researchers looked at East Asia, where Rh negative blood is genuinely rare, well under 1% in most populations, so rare that in some places a hospital may go years between cases. And there, in Japanese donors who tested Rh negative, something else again. In more than a quarter of them, the Rhd gene was present and looked intact. Present, apparently whole, and yet producing nothing on the surface of the cell. Sit with that for a moment, because it is the single most important fact in this entire subject, and almost nobody discussing Rh negative blood online will tell you about it. Rh negative is not one thing. It is at least 3 separate genetic events in 3 separate populations on 3 separate continents, arriving independently at the same outcome. In Europe, the gene was deleted. In Africa, the gene was silenced from the inside. In parts of Asia, the gene sits there in a state we still don't fully understand. If you are Rh negative, the reason you are Rh negative depends entirely on where your ancestors came from. There is no single Rh negative lineage. There is no one bloodline. There is a shared outcome produced by unrelated accidents, which is a very different thing, and in some ways a far more interesting thing. And there's one more layer underneath all of this, which almost sounds like a philosophical joke. The Rhd gene has a twin next to it, a partner gene called Rhce, which produces the other markers in the rhesus system. These 2 genes are similar enough that we know one is a copy of the other. Long ago in our primate ancestry, there was a single original gene, and at some point it duplicated. The copy drifted, took on a new job, and became Rhd. And here's the part that catches people. The original ancestral gene at that position no longer exists in our species, not in anyone. What we all carry now is the aftermath of a copying event, one gene that stayed close to the original role, and one copy that became the D antigen, and then, in some people, got deleted again. Duplication, then loss, the genome writing something, revising it. And applied here, the idea goes like this. You can carry 2 deleted copies and be Rh negative. You can carry 2 working copies and be Rh positive. Or you can carry one of each. You'd test as Rh positive, and you'd never know the difference from a blood card. And the hypothesis is that this middle group, the carriers, have some measurable edge that the other 2 groups don't. There is research supporting it, and it's worth describing carefully. One study examined more than 2,500 people, working out their underlying genetic combination from their own blood type and their parents' blood types, and then comparing physical and mental health measures across the 3 groups. The finding was that Rh negative individuals scored worse on a range of health measures than Rh positive individuals overall. But more importantly, the carriers, The ones with one of each version came out ahead of both of the other groups. Better than the fully positive, better than the fully negative, exactly the pattern the theory predicts. A related line of research found something even more specific. When people were latently infected with a common parasite called Toxoplasma gondii, which quietly infects a large fraction of humanity and is known to slow reaction times, the carriers held their reaction times far better than either of the other groups, as though the middle position confers a kind of resilience, not to any one disease, but to the general burden of being an organism in a world full of things trying to get inside you. Now, honesty matters here, so let me be straight about the state of this. Much of this work comes from a single research program, led by a Czech evolutionary biologist named Jaroslav Fleger, and while it has been published in serious journals and pre-registered to guard against cherry-picking, it has not been broadly replicated by independent teams. The effects are statistical, not dramatic. They tell you nothing about any individual person. If you're Rh negative, this research does not mean you're sick, weaker, or destined for anything at all. Plenty of Rh negative people live long, healthy, entirely ordinary lives, and the differences described are averages spread across thousands of people. What the work does do is offer a plausible mechanical answer to a question that had no answer for 80 years. And it may turn out to be right, or partly right, or replaced by something better. That's how this works. But even setting the mechanism aside, the raw geography of Rh negative blood tells its own story. And this is where it stops being medicine and starts being history. Here's the global picture. Roughly 15% of Europeans and people of European descent are Rh negative, around 8% of African and African American populations. In East Asia, well under 1%. Among indigenous American populations, it approaches 0. Before European contact, it may have been effectively absent altogether. Those are enormous differences. Blood types don't usually vary that sharply across the world. And then you go to one specific place, and the numbers become genuinely startling. In the Western Pyrenees, along the border between Spain and France, live the Basque people. Among them, Rh negative blood runs at somewhere between 30 and 35%, more than double the European average, and the highest concentration measured anywhere on Earth. When researchers count the underlying gene copies rather than the phenotype, the Basque figure reaches into the high 40s. Nearly half the relevant genetic material in that population is the deleted version. There's a secondary peak too, less famous, among Berber populations in the Rif and Kabylia regions of North Africa, where the frequency sits somewhere around 15 to 20%. These are not random hotspots. And for decades, nobody could say why they existed, which left a vacuum, and that vacuum got filled with some remarkable stories. But then ancient DNA arrived, and ancient DNA has a habit of quietly ending arguments. Over the last 15 years, researchers have learned to extract and sequence genetic material from ancient human remains, from skeletons thousands of years old, from teeth and the dense bone of the inner ear. And by doing that across Europe, they've built something like a layered map of who lived where and when. Modern Europeans are a blend of 3 main ancestral populations. First, the hunter-gatherers, the people who were here during and after the last ice age, who lived by hunting and foraging for tens of thousands of years. Second, the farmers who arrived from the Near East, beginning around 9,000 years ago, bringing agriculture with them. And third, pastoralists who swept in from the steppe grasslands to the east in the Bronze Age. Almost everyone in Europe today carries some proportion of all 3. And when researchers examined the rhesus gene across these ancient groups, the pattern was striking. Among the Neolithic farmers coming out of the Near East, Rh negative frequency was very low, on the order of 4%. Among the Western hunter-gatherers, the Ice Age population, it was around 24%, 6 times higher. Read that again because that is the secret in your DNA. Rh negative blood is, to a substantial degree, a marker of Europe's oldest layer. The hunter-gatherers, the people who painted caves, the people who followed herds across a cold continent, who lived through the last glacial maximum in the southern refuges where the ice never quite reached and who were largely absorbed, replaced, and diluted when farming arrived and populations exploded. Their genetic contribution to modern Europeans is real but modest in most places, but the deleted rhesus gene came through them, and where their ancestry stayed concentrated, so did it, which explains the Pyrenees perfectly. The Basque people are a well-documented genetic isolate. Their language, Euskara, is unrelated to any other living language on Earth. Not Indo-European, not related to anything spoken nearby. A genuine linguistic island. Their genetics show comparatively little of the Bronze Age steppe input that reshaped the rest of the continent. They sat in mountain valleys, in a corner of the world that migrations tended to flow around rather than through, and they held on to an older configuration for a very long time while everywhere else was being mixed and remixed. So a trait that was common in Ice Age Europe stayed common there, while it was diluted everywhere else. Not magic, not mystery. Isolation plus time, which is the most powerful combination in population genetics. Now, no honest treatment of this subject can avoid the other explanations that have circulated for decades, so let's deal with them directly. If you've searched this topic before, you have almost certainly run into the claim that Rh-negative blood comes from somewhere outside the human family, that it's a trace of extraterrestrial contact, or of the Nephilim from ancient texts, or of interbreeding with Neanderthals. These ideas are old, persistent, and honestly, you can see why they took hold. The distribution is genuinely strange. The Basque language really is an orphan. The evolutionary paradox was real and unresolved for decades. When the experts have no answer, other answers move in. But we can test them now and they don't survive contact with the data. The Neanderthal version fails first. We have sequenced Neanderthal genomes in detail from multiple individuals, and the rhesus story in those genomes does not support the claim that our Rh-negative deletion came from them. And the non-human origin claim fails on something even simpler. The thing we established at the very beginning of this: if Rh-negative blood came from one exotic source, whatever that source might be, it would leave one genetic signature. It would look the same everywhere. It doesn't. In Europe, it's a deletion. In Africa, it's mostly a silenced gene sitting exactly where it belongs. In parts of Asia, it's a gene that's present and unexplained. That's not a fingerprint. That's 3 separate fingerprints that happen to produce the same smudge on the glass. A single origin cannot produce 3 mechanisms, and 3 mechanisms is what we find. The truth, as usual, is stranger and better. Your blood type isn't evidence of a visitor, it's evidence of an ancestor, a specific human mortal ancestor who lived somewhere in Ice Age Europe, whose chromosome folded wrong one day, and whose descendants somehow did well enough for that error to travel 40,000 years and arrive on a card in your wallet. There's one more thread worth pulling, and it goes to a question people rarely ask. What was the D antigen for in the first place? Why does the body build it at all if a substantial share of humanity gets along fine without it? The rhesus proteins aren't decorative. They're part of a complex embedded in the red blood cell membrane, working together with a partner protein produced by a gene called RAG. That complex appears to be involved in moving ammonia across the cell membrane in both directions, a housekeeping job, part of the constant chemical management that keeps a cell alive. And beyond the transport work, the complex plays a structural role, part of the scaffolding that gives a red blood cell its shape and its flexibility, which matters enormously for a cell whose whole career is squeezing through capillaries narrower than it is. We know this because of what happens when the whole system fails. Missing the D antigen alone, as Rh-negative people are, causes no health problem whatsoever. The rest of the complex carries on, and the body doesn't miss it. But there are people, extraordinarily rare people, who are missing every antigen in the rhesus system. All of them. Their red blood cells are, in rhesus terms, completely bare. The condition is called Rh null, and it's usually caused by mutations in that partner gene, RHAG. And those people are not fine. They live with a chronic, usually mild hemolytic anemia because their red cells are fragile and break down faster than they should. Some develop a red cell disorder called hereditary stomatocytosis. The scaffolding, it turns out, was load-bearing after all. Our null blood has a nickname. They call it golden blood, and there's a good reason for it. In the entire recorded history of blood banking, somewhere around 43 people have ever been identified with it worldwide, across all of human history and all of modern medicine. Current estimates put the odds at something like 1 in 6 million. There are fewer than 10 active donors on the planet. The first case was found in 1961 in an Aboriginal Australian woman, and here's the detail that tells you how unexpected it was. Before she was